of platelets and their relationship to blood clotting or hemostasis, a more general term, prevention of blood loss. And very briefly last time talked about vasoconstriction and narrowing of the blood vessels, which will help, of course, to reduce blood flow down the area. The vessels are narrowed, less blood is flowing, and therefore less blood is lost. I also mentioned the platelet plaque and how it is formed. Remember, the platelets normally don't stick to the inner lining of the blood vessel, but when it's damaged, the collagen is exposed, and through von Willebrand's factor, the platelets stick, they adhere, the 3 A's, they adhere, they become activated, change their shape, release various substances from their granules, and these substances in turn help in vasoconstriction, and in making the platelets stickier, and they stick to each other, they build up, until you have a platelet plug. It just says literally plugs a hole in the vessel wall. But I also said last time that these two first steps are not always enough in themselves. If it's a small bit of damage, then maybe they're okay. But particularly if we're talking about a high pressure blood vessel or large amount of damage, these two in themselves are not enough. And we have to move on to the formation of a definitive blood clot. And so what I'm going to be talking about for the rest of today's lecture is really number 3 here, secondary hemostasis, formation of a blood clot, blood coagulation, and asking these questions. First one, I've more or less answered it. Why is it necessary? Because the first two aren't really enough in themselves. The platelet plug is a fairly delicate structure, and particularly if there's a large amount of blood flow or high pressure, it can get washed away. So you need to have something that's firmer and will last for longer. What is involved in blood clotting? That's a question that covers a lot of ground. The short answer to that, what is involved in blood clotting, is the formation of something called fibrin, an insoluble protein which traps the various blood cells within its network. It forms a mesh or a network, and it traps the blood cells and stops the blood from being lost. So really, another way of saying what are we going to talk about today, at least a large part of it, is how do we form fibrin. And this is what I'm going to be coming back to. What does a blood clot look like? You can look in just about any textbook and you'll see a figure which looks something like this, blood clot. And I've just highlighted here in the yellow, these are the fibrin threads, this insoluble protein, and trapped in this dense network or this mesh are red blood cells, platelets. You can see them a bit more clearly here, the green ones. This particular figure doesn't seem to have, I don't think, any white blood cells in it, but they'd be around there somewhere. Of course, red cells are much more numerous, so you see more of them, and also the platelets. So what I'm going to be asking next then is, how does all this happen? How is fibrin formed? And the simple answer to that is shown in figure 59, like the summary of blood clotting. And this is something where we need to start, or at least I think it's more useful to start at the end and work backwards. This is what we're trying to produce, fibrin, insoluble protein. And fibrin is derived from fibrinogen, which is one of the main plasma proteins. Fibrinogen is a soluble protein, and it's converted to the insoluble form by this crucial, very central enzyme, central to this whole story in blood clotting, called thrombin. If you just think about that statement for a moment, you realize that thrombin cannot be present in an active form all the time. Because if it were, then all of the fibrinogen that you have everywhere in your plasma would continually be converted to fibrin. And most of the time, you don't need that. Certainly don't need it, it could be very dangerous. So thrombin has to be present in an inactive form, prothrombin, most of the time, and then it needs to be converted to the active form thrombin only when and where you need it. And what converts prothrombin to thrombin, put here as a star standing for something called prothrombinase. I put that in inverted commas because it's not a single entity. It's a complex of several different substances, and we'll see what those are in a moment. But that just sort of pushes the story back one step. Prothrombinase cannot be there all of the time. Otherwise, you get continual production of thrombin, continual production of fibrin, and that will be what you want. So we have to explain how prothrombinase is produced only when and where you want it. And we could expand this scheme upwards and say, well, something produces prothrombinase, that something has to be activated only when and where you need it. Go back another step, say the same thing, and this story's got to start somewhere. We can't go back forever. So what I want to try and show you is how this actually does start, and how you get this sequential activation through this cascade or waterfall system that ultimately brings about the formation of thrombin and hence of fibrin. And you need to just know or become familiar with a few names here, but I would say straight away that unless you're going on to work in this field or doing other stuff, you don't need to necessarily remember these. But you'll probably, in your reading, come across some of these names. And these are the plasma clotting factors. Well, we've already come across some of them. Fibrinogen. And the ones I put in capital letters there are usually given their names. Other ones that are not in capitals are sometimes called by their names, but more often may be called by Roman numerals. And these Roman numerals were given to try and simplify this whole story. I'm not sure if they were successful in doing this. They're numbered not in the order in which they play a part in the formation of a clot, but in the order in which they were discovered. So it doesn't really help you all that much in some ways. Prothrombin, or factor 2, we've already mentioned. Calcium, we don't go around talking about plasma clotting factor number 4, you talk about calcium, and so on. Some of these are rather odd names, Hardeman factor, Christmas factor, and so on. And these are named after some of the patients that were first shown to have problems with certain clotting factors. And this Christmas factor, just an interesting aside, was named after a young boy at that time called Stephen Christmas. Somewhere in Canada, I'm not quite sure where it was, somewhere down east, Montreal, Toronto, I think. And he was lacking this particular factor, now named after factor number 9. And lacking factor number 9 gives rise to a type of haemophilia, not the one that's most common, but the second type. And it says that Stephen Christmas, all his life, he fought for help for people who need haemophiliacs who needed treatment. And there are many coincidences surrounding Christmas that haunted him. The article that announced the discovery of the Christmas factor was actually published in the 1952 Christmas edition of the British Medical Journal and met with some negative reactions. People said, should a disease be related to the image of Christmas? To which the authors answered that they promised that if they found a precursor protein to this Christmas factor, they would not call it the Christmas Eve factor. Sadly, there's a sad story, this really, that Stephen Christmas, he died at the age of 46 from HIV, which he contracted through treatment with tainted blood products. And he died just 5 days before Christmas in 1993. So a bit of human interest there. Well, what do we need to know about these factors? Many of them are produced in the liver. Many of them are proteins, well, obviously not calcium and But many of these are proteins which are produced in the liver. And the ones that I've put a star by require vitamin K for their synthesis. And the reason why I'm telling you that will become apparent later on. So the question then is, how can we fit those factors into this blood clotting story? I mean, you know, this is an honours class, you need something more than this. But how much do you want or need? And I'm just going to very quickly put up this figure, and I'm doing it very quickly for two reasons. One, you know, we're supposed to give attributions to say where we found it. I can't find where I found it. So I'm not supposed to really have it up. Anyway, it's not mine, obviously. I'm not claiming it's mine. But I just wanted to show you, I'm not sure if this is the whole story. You don't need to know this amount of detail. But why do I show it to you? Because I think if you see something like this, with arrows and factors and things all over the place, all inside the blood vessel to form a blood clot, when you look at what I do want you to know, you say, oh, that's not so bad. You know, it could be worse. So let's look at the sort of a medium amount of information. You need more than the simplest one that I showed you initially. You don't need that one I just printed off there. But I think this is somewhere in between. So what this is showing is what I'm calling the traditional mechanism for blood clotting. And again, I'll tell you why I'm calling it that in a moment. So here at the bottom, which I've just outlined in the blue, is basically the story you know already. This is called the common pathway. And you absolutely must know this one. Prothrombin in the presence of prothrombinase, which is actually all of these things within this circle here, is converted to the active thrombin. Thrombin converts soluble fibrinogen to fibrin, which initially is not very stable, but then there's a final step added here where fibrin becomes stable, forms those threads, traps the platelets, traps the red cells, and you've got a good blood clot. So now we can ask the question, well, where does prothrombinase come from? This mixture of factor 10, 5, calcium, PL, phospholipid present on platelet surfaces. So we're going to come back a bit here to platelets. They play a role not only in forming a plug, but also in the formation of a real blood clot. So how do we form this complex here? Well, there are two pathways. There's a so-called extrinsic and a so-called intrinsic pathway. Let me just briefly tell you what these are about. If you take a sample of blood out of somebody, let's say you're an absolute expert in doing this, you've got a very fine, sharp needle, you can get into a blood vessel, extract some blood without causing damage, take that blood, put it in a test tube, put it on the bench, and wait. What's going to happen? It's going to clot. Within a few minutes, that blood will clot. But when the blood was in the body, it didn't clot. It was flowing. What have you done? Have you added anything to the blood? No. Have you taken anything away from the blood? No. The blood is still the same blood. All you've done is take it out of the body, put it in this tube. Well, it's thought that in that situation, what's triggering the formation of the clot is the contact with this foreign surface. The glass of the test tube is foreign. It's not the usual surface that the blood encounters. There's something about that foreign surface which can trigger the so-called intrinsic pathway, contact activation, contact with the glass. This is called the intrinsic pathway because everything you need for the blood to clot in that test tube is already present in the blood itself, is intrinsic to the blood. And what happens is that when it contacts the glass, factor 12, hardening factor here, so you don't need to remember that, is activated to active 12 subscript A, means it's in an active form, and then active number 12 activates number 11, and then active 11 in the presence of calcium activates 9, 9 in the presence of more calcium, platelets, platelet phospholipid, and factor 8, active 8, all of these things together will activate factor 10 to active 10, and then active 10 to 5 in calcium and phospholipid activates thrombin, and away you go and form a blood clot. Now, I know you're wondering, do we have to remember all of this, exact numbers and things? That to me is not the major thing. If you want to find, if it so happens that it's 6 in your mind, that's great. But the more important thing is to understand the concept here, rather than remembering whether 9 comes before 11 or after 10 or whatever. So it's the idea of this sequential activation. What's the significance of that, Keisha? Having sequential steps? So you don't form a clot accidentally. There's, I suppose, more opportunity for control at each step. Anything else? Yes? Amplification. Yes, I think we talked about another cascade system somewhere, and same idea that at each step you can get amplification. So you can start with a very small number of molecules of factor 12 being activated, and then so on each step more and more, and you end up with a good clot. But this is a rather artificial situation. What about in real life when there is damage? And this is thought to, according to this scheme, trigger the extrinsic pathway. An extrinsic refers to the fact that something is added to the blood when there's damage. And this something is called tissue factor, yeah, there. So when tissues are damaged, tissue factor becomes added to the blood, and in the presence again of calcium, which seems to be needed at many steps, platelet phospholipids provide another cofactor and an active factor 7. This whole complex is another way of activating factor 10. And so in this shorter extrinsic pathway, you've again activated factor 10, you form the prothrombinase complex, and thrombin and so on. Okay, so what's wrong with this? Isn't that good enough? Well, more recent work has shown that we need to make some modifications to this. The basic story is still there, but there are some things important things that need to be somewhat changed. And the bits of information that are relevant to this have been seen in people who have deficiencies in certain factors, and see what happens. And you don't need to remember these ones, but just the idea that if you have factor 7 missing, then this person will show severe bleeding. And you say, well, why? I mean, if this extrinsic pathway is not working, you know, this traditional scheme looks as if we've got a backup. Why can't we then use this pathway, the intrinsic one? If factor 8 is missing, which is in the intrinsic pathway, you could say, well, that's okay, it's not good, but we can use the extrinsic pathway. But it doesn't seem to work like that, because either one of those, if it's missing, you've got severe problems, which suggests that there's interaction between those two so-called intrinsic and extrinsic pathways, and you need both of them to be functioning properly in order for proper blood clot formation. On the other hand, if you're missing factor 11 up here, okay, there are some problems, moderate, but if you're missing factor 12, hardening factor, way at the beginning here, in vivo, meaning in the whole body, no problem. In a test tube, yes, but you know, we're not really quite so interested in what happens in a test tube as what's happening in the real person. So factor number 12, which we've just said in this traditional scheme, initiates the intrinsic pathway, to factor 12 does not seem to be necessary. So using those sorts of results of natural experiments and so on, we can modify the blood clotting scheme. to something like this. So again, we have this common pathway in blue at the end. Nothing much has changed here. Prothrombinase is complex, activates thrombin, forms fibrin, stay unstable and then stable. But what is somewhat different here is that we can now add that once you have formed a little bit of thrombin, this can feed back. So what about these so-called extrinsic and intrinsic pathways? Are they still valid, or do we still use them? Sort of. And let me explain that. What is now believed to happen, okay, in this previous scheme, the difference between intrinsic and extrinsic is more or less a case of where is this happening. If it happens in a test tube, it's the intrinsic pathway, the green one. If it's happening in the body, it's the extrinsic one, and never the two to meet until they come to the common pathway down here. What people now think is happening is that it's more a question of timing rather than location. So what they think happens is that inevitably there's going to be some tissue damage. I don't think you could have blood loss without some tissue damage. And this damage provides the tissue factor and all the other factors, and this is what initiates clotting. And this pathway then leads into the common pathway, and you get a small amount of thrombin. And that amount of thrombin is enough to get things going, and it's also enough to cause a feedback which will, through what we used to call the intrinsic pathway, will now amplify even more thrombin formation. So once you've produced a small amount of thrombin because there's been some tissue damage through the old extrinsic pathway, now it's called the initiating pathway, you get small amounts of thrombin. And then all these dotted arrows that I've added in show you that thrombin not only causes fibrin formation, but it can activate factor 5, factor 8, factor 13, maybe some other ones too, and it can cause factor 11 to be activated. So you don't need factor 12 in a real-life situation. So again, the emphasis is on initiation through the red pathway, forming small amounts of thrombin, feedback, amplification, bringing into play the green pathway, and then more thrombin form and more clot formation. So this sort of explains why you need both pathways. And this can also be added to by saying that there are arrows which go across from one pathway to the other, and just showing 1 here, that 9 can be activated to active 9 by this complex here, which normally we thought of as being part of the red pathway. 9 is the green one. So there's arrows going from red to green, other arrows which I haven't even put in, because this is where it starts to look like that really complicated one, going from green to red, backwards and forwards, a whole spider's web of interactions between the two pathways. Let's just summarize that then. Thrombin is really the key to all of this, the center there. What does thrombin do? It activates platelets. We saw that in the figure last time, where we had when plaque platelets are activated, phospholipids become exposed. Phospholipids, as we've now seen, are part of the story in the production of thrombin. Once you've got thrombin, it can feed back and activate more platelets. Thrombin converts fibrinogen to fibrin, it forms a clot. And it also activates other clotting factors, the dotted arrows shown here. And I think that's number 4 we're coming to a bit later. So what I hope you can get then is the general idea of how a blood clot is formed. thinking about the reasons for it, the general mechanisms, what's involved, and not worrying too much about the exact numbers of the different factors and the order in which they appear. Except the common pathway, you should certainly know that one. So how is all this regulated? I mean, you don't want this just to be an explosive event and clots being produced left, right, and center. You have to have some control. So you have to prevent clot formation when you don't need it. So we need to talk a little bit about anticoagulants. And we also need to talk about the breakdown of clots, because you don't want them to last forever. If you have damage in a wall, you want a clot right now as soon as you can. You want it to last for a while, days maybe, and then as the wall gets repaired, you no longer need to have the clot, so you want to break them. Let's look briefly at those anticoagulants first of all. And these can be divided into naturally occurring ones and also the ones that are used clinically. So this figure here, number 66, just names a few of the naturally occurring ones. And again, don't need to remember details here, but there is something called tissue factor pathway, which is this red one here. There's an inhibitor there, which inhibits these particular factors here. There's something called antithrombin 3, which, as the name tells you, it inhibits thrombin. So if you're getting too much thrombin, then you've got a naturally occurring break for this system, just to slow things down if you're getting more than you need. You've got something called thrombomodulin. I'm going to talk about this because this is, I think, very interesting, very crucial. And also protein C and F. So this says that thrombomodulin changes thrombin activity. What can that mean? Well, I've been emphasizing many times that thrombin is essential for clot formation. It's procoagulant. And that's exactly what you need when there's damage. But what happens when you form thrombin, and maybe it spills over in the bloodstream and it travels downstream, and you've got a normal part of the blood vessel, you don't want thrombin to be causing clotting in that normal blood vessel. So it's almost like magic. Thrombin can become an anticoagulant. Mostly you talk about thrombin, you hear about it, you think about it as being something which forms a blood clot, but it can also have the opposite. And this is shown here. So the first point is that thrombin, as I've just been discussing, favors clot formation. Okay? However, if you talk about a region of normal blood vessel lined by normal endothelial cells, they haven't been damaged, they're just perfectly normal cells, you don't want a clot in this region. These normal endothelial cells have, or express on their cell surface, this molecule called thrombomodulin. And thrombomodulin combined to thrombin. And when that happens, when thrombin is bound to thrombomodulin in a normal part of the blood vessel, it somehow, and I don't know all the details, but somehow it switches the activity of thrombin to being anticoagulant. And the way in which it works is that this thrombin, but only if it's bound to the thrombomodulin, can activate something called protein C, which is yet another protein in the plasma, normally in an inactive form, and it becomes activated in the presence of thrombin bound to thrombomodulin. And this active protein C, in the presence of cofactor protein S, in the presence of protein S, it causes a breakdown of factors 5 and 8. 5 is here, 8 is there, And if you don't have these factors, then clotting is inhibited, because you need those factors to produce a normal blood clot. So this is a very clever scheme, very clever arrangement, where you can switch the action of thrombin. It becomes diametrically opposite to what you think of as being its normal function. Yeah? Okay, the question was, does the binding of thrombin to thrombomodulin, as well as causing activation of protein C, does it also prevent the feedback of thrombin, causing amplification in the pathways that we talked about? Just trying to think whether it would. As far as this is concerned, there shouldn't be any of that in this region, the blood vessels going on. I'm not actually sure if you put it in there in a bound form, what it would do. I'll have to think about that. But I think in a real life situation, it's not likely that the thrombin that's in this region would be causing that feedback in the first place. But if it were, whether it would be affected, I don't know. Okay, so do you get the general idea then of this ability of this one and the same enzyme, the thrombin, to have opposite action? Okay, still sticking with anticoagulants, you asked the question, what use can we make of this information in a clinical situation? And I think this is interesting and important to know, because it helps you to see, did you understand how clotting works? Which you need to in order to understand how you can prevent it. And one anticoagulant, type of anticoagulant that can be used, are calcium chelators. We only need to look at this briefly and see calcium, 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 might be other places too. Calcium ions are essential for blood clotting. If you chelate the calcium's still there, but it's not present in the ionized form, which is necessary to play its role there, then blood will clot. You should only use that type of anticoagulant in vitro. Why? Why would you not want to do that in vivo? In vitro is literally in glass, in the test tube, out of the body. In vivo is in the living body. Why would you not want to use calcium chelators in the body? Would they work, first of all? Would they prevent clotting? Yes? Yes, please.
Isn't calcium an important signaling, or calcium plus important signaling molecule for a lot of other processes?
Exactly, yes. Calcium, ionized calcium, is absolutely crucial for many other processes to do with nerve, muscle, transmitter release, all, long list of why you need ionized calcium. So these chelators would work. They would prevent blood clotting in vivo, but you don't want to do that because everything else, you'd stop the blood clotting, but you interfere with so many other normal processes. What you can use in vivo and in vitro is a substance called heparin, which potentiates the effects of the naturally occurring antithrombin, which prevents the action of thrombin and other factors too. And this is something that's, you inject the heparin and it works almost instantaneously, and it will last for, well, it depends how much you give, but it lasts for maybe a few hours, and then you can give some more. The third one, we come back now to vitamin K. Antagonists of vitamin K prevent blood clotting. And the way in which they do it, they inhibit the synthesis of certain of the blood clotting factors. Those are the ones that were starred on that earlier figure. And so if you don't have the factors, which are made in the liver, the blood won't clot. And you can only use this one in vivo. This one's not going to work in vitro. I would like to ask you why, but I've already told you the answer, because it's inhibiting synthesis of proteins, which obviously only occurs in the living body, it doesn't occur in the testes. So it takes a while, it takes a few days for this to exert its action, but then it will be effective. And an example of something that's used here is the substance warfarin. You may have heard of this. It's a dicoumarin derivative, and it's used in rat poisons. And for a long time, I used to think that this, because it was a thing that killed off rats, I thought the name came from something like, warfare, fighting the rats. Well, that's actually not the case. It stands for the name of the place where it was discovered and worked on, which is the Wisconsin Alumni Research Facility, something like that. And it's an interesting story. Back in I think the late 1920s and the 1930s, quite a lot of farmers were finding that their cattle, other animals, were dying, were bleeding to death. And apparently one snowy morning in February 1933, this particular farmer in Wisconsin went into somebody's laboratory there at the university carrying a milk can full of blood that refused to coagulate. And outside he had a small heap of spoiled sweet clover hay and a dead cow freezing in the back of his truck. And he said, help me. So what's going on? It's the key there, I think, is this spoiled sweet clover that they were using as feedstuff for their animals. And eventually extracts were made and it was shown that this dicoumarin derivative was present in this spoiled feedstuff. And you think, well, why did they keep feeding it to their animals? Well, just think of the timing. This was the 1920s, 1930s, time of depression. They couldn't afford to lose the cattle, but on the other hand, they couldn't really afford to just throw away this spoiled food stuff. So it was out of this very bad situation that this anticoagulant was discovered, and apparently is the most commonly used anticoagulant in the world. So in the UK, it's estimated that at least 1% of the population and 80% of the over 80s are taking this anticoagulant regularly. So again, an interesting background to that. Okay, so that's just a little bit about how to prevent the formation of a blood clot when you don't want one, either in the body or in a clinical situation if somebody's undergoing certain procedures and you don't want the blood to clot while they're having a certain operation or something, you can use various drugs. But what about going back to when you do have a blood clot formed, normally, in the right place at the right time, and then healing occurs and you need to get rid of it. And so the process of breakdown of a blood clot I also need to understand. And this is a process called fibrinolysis, the breakdown, the lysis of fibrin. So what we're trying to do here is to remove this insoluble fibrin that has been so carefully produced under the blood pathways shown previously. And the key enzyme in this breakdown is the enzyme plasmin. So for forming a blood clot, you absolutely need thrombin. For breaking it down, you need plasmin. And plasmin is produced from an inactive precursor, plasminogen, under the influence of plasminogen activators. So it's another example of a cascade system, but it's a shorter and somewhat simpler one than the formation of the clot. We still need to ask, well, what are these plasminogen activators? And I've just listed in the boxes here some of the naturally occurring ones. First of all, something called TPA, tissue plasminogen activator, which comes from various tissues, normal endothelium, most tissues, and things like exercise and stress increase the release of this. So, various reasons exercise is good for you, and in that long list of reasons why it's good, here's another one to add, that it increases the release of this, or at least good in most circumstances. You get a breakdown if clots have formed and you don't need them, then the exercise will help you to break them down and presumably wash them away because blood's flowing fast. Using this knowledge, people have produced what they call clot busters, which you may have heard of. These are thrombolytic drugs. A thrombus is an unwanted blood clot, and this is what they're trying to break down. And here's one example. I think there's probably some maybe newer and better ones now, but tenecteplase was one of the first ones that was brought in. And the idea here is that when an alarm goes out, somebody's had stroke or heart attack, and the ambulance rushes there to pick them up to take them to the hospital. If the paramedics in the ambulance are supplied with these clot busters, if it's appropriate, they can start treatment straight away before, it might be a few hours before the person goes into the hospital and can be seen there. And it's very important to get in quickly. These things don't seem to work all that well as time goes by, but getting, I don't know what the time limit is. Anybody know? It's maybe a couple of hours or something like that. The quicker the better. And you can then break down clots before they've got so big that they're preventing blood flow completely to the brain or a region of the heart. Okay, and this produces the activated plasminogen plasmin, breaks down the fiber into a soluble group of molecules, degradation products, which can then be washed away. And then in real life, it just said that there are various balancing factors in a normal situation to keep things as they should be, to keep things optimal. And then the final figure in this section is just to give you a few things to think about, really. What can you do